So now we will be discussing nutrition in bacteria. Two categories are there, that is autotrophs and heterotrophs. Bacteria is nutritionally divided into the two categories, and this differentiation is on the basis of carbon source which is utilized by the bacteria. Now the autotrophs, they have the ability to use inorganic carbon. Yes, they can use inorganic carbon compounds. That is carbon dioxide which is present in the environment. Now the heterotrophs, the ability to use inorganic carbon that is absent, they can use only organic compounds. They can use organic compounds. So you know that these organic compounds, they are already formed by some other organisms. They are made by some other organism. It means they are dependent on other organism for the carbon requirement. They are dependent on the other organism for the carbon requirement. That's why they are known as heterotrophs. Right? So if we talk about the autotrophs, they use inorganic carbon, and heterotrophs, they can only utilize organic carbon compounds. Now first of all, we will be dealing with the autotrophs here. Now the autotrophs, they fix carbon dioxide and results in the production of food. कार्बन डाइऑक्साइड को फिक्स करेंगे और उससे फूड फॉर्म करेंगे. Now for the fixation of carbon dioxide, there is requirement of energy. If the energy utilized by the bacteria is light energy, for the carbon dioxide fixation, if light energy is utilized, you call this category of bacteria as photoautotrophs. But for the carbon dioxide fixation, if chemical energy is utilized, you call them as chemoautotrophs. So two further categories are there. For the synthesis of food, if light energy is used, so you call this category as photoautotrophs. And if chemical energy is utilized, then chemoautotrophs. Under the photoautotrophs, two categories I will be dealing here. One is PSB, that is purple sulfur bacteria. Another one is GSB, that is green sulfur bacteria. In the case of the purple sulfur bacteria, the pigment which is involved in the light absorption, or you can say the light absorbing pigment which is present, that is bacteriopurpurin. Yes, so this is purple color pigment present in the bacteria for absorbing light energy. Now in the case of the green sulfur bacteria, it is bacterioviridin. It is also known as chlorobium chlorophyll. So these are the pigments which are present in the bacteria, and you know this very well. The pigments which are present in the plant for absorbing light energy, that is chlorophyll. Now coming to the photosystem. Photosystem represents a group of pigments. Yes, that is group of pigments which are involved in the photochemical act. Both in the purple sulfur and green sulfur bacteria, there is presence of only one photosystem. Right? Now, in the case of the purple sulfur bacteria, the photosystem which is present, it is activated by absorbing the light energy of 890 nanometer. I am repeating this. The photosystem which is present in the purple sulfur bacteria, it is activated by absorbing the wavelength of light, that is 890 nanometer. And in the green sulfur bacteria, it is activated by absorbing the wavelength of light of 840 nanometer. So this is the condition in the bacteria. Now we will compare this with plants. So just now I have told you that in the bacteria there is presence of only one photosystem, but in the plants there is presence of two photosystems. Yes. So photosystem 1, photosystem 2. This is how you represent it. Now photosystem 1, it is activated by absorbing the wavelength of light of 700 nanometer, and photosystem 2 by 680 nanometer. Now the photosystem 2, when it absorbs light energy, it results in breakdown of water. Yes, photosystem 2 light energy absorb करेगा, जिसकी वजह से water का breakdown होगा. Yes, you call this as photolysis of water. Due to the breakdown of water, there is a release of hydrogen ions, electron, and oxygen. Now the electrons which are released, these electrons, they enter ETS, that is electron transport system, and through the electron transport system there is production of ATP and NADPH. Now the ATP and NADPH which is produced here, that is used for carbon dioxide fixation, which in turn results in the synthesis of food. Are you getting this sequence? In the plants there is presence of two photosystems. Here, photosystem 2 is involved in the splitting of water, which results in the production of electron. Now, these electrons, they enter ETS, which will produce ATP and NADPH, which in turn are utilized for carbon dioxide fixation, resulting in the production of food. So you can see here that in the case of plant, the electron and hydrogen donor which is there, that is water. Very important point to note down here. The electron and hydrogen donor in the case of plants, that is water. If we look into the bacteria, if we look into the metabolism of bacteria, in the bacteria, there is only presence of one photosystem. The splitting of water in the bacteria is absent. If the splitting of water is absent, it means water will not be used as electron and hydrogen donor. Here the electron and hydrogen donor, they are compounds other than water. In the case of the purple sulfur bacteria, the electron and hydrogen donor is sodium thiosulfate, right? And in this case, it is H2S. So you can see in both these cases, the electron and hydrogen donor, they are sulfurous compounds. They are sulfurous compound. That's why we are calling this category of bacteria as sulfur bacteria. Right? Now the electron and hydrogen donor which is utilized here, that is inorganic. Yes, we are now not talking about the carbon source. We are talking about the electron and hydrogen donor that is also inorganic. Yes, so when electron and hydrogen donor, that is inorganic compounds, then you call this category of bacteria as lithotrophs. So it means we can say that photoautotrophs, they are photolithoautotrophs. Right? Yes. Why we have called them as phototrophs? Because they have the ability to utilize light energy. Litho, because the electron and hydrogen donor utilized is inorganic compound. And autotrophs, because they can use inorganic carbon. Right? Now, in the case of plants, what we have seen? During the photosynthesis, there is splitting of water. Due to the splitting of water, there is release of oxygen. It means in the case of plants, when photosynthesis will occur, it is accompanied by the release of oxygen. Now, this type of photosynthesis, you call it as oxygenic photosynthesis. But if we talk about these bacteria, in these bacteria, now utilization of water is absent, oxygen release is absent, so this is known as anoxygenic photosynthesis. So let's write some of the important differences between the bacteria and plants. Now, in the case of bacteria, there is involvement of only one photosystem. In the case of plants, two photosystems they are involved. In the case of the plants, the electron and hydrogen donor, that is water. And in these bacteria, sulfurous compounds, they are used. Due to the splitting of water, there is release of oxygen. That's why you call it as oxygenic photosynthesis. And here oxygen release is absent, so that's why this is anoxygenic one. Right? So this is all about the first category of the bacteria, that is photolithoautotrophs. So now I will be starting with the second category, that is chemoautotrophs. To explain chemoautotrophs, I will be citing example of nitrifying bacteria. Nitrifying bacteria is present in the soil. Now these bacteria, they convert ammonia into the nitrite and then nitrite into the nitrate. They are involved in the conversion of ammonia into nitrite and nitrite into nitrate. At the end, there is synthesis of nitrate by these bacteria. That's why this category of bacteria is known as nitrifying bacteria. Now, at this step of the nitrification, there is involvement of nitrosomonas and nitrococcus. These two bacteria, they are involved here. At the second step, there is involvement of nitrobacter and nitrocystis. Now tell me, this reaction is a oxidation reaction or a reduction reaction? Yes, there is addition of oxygen. So surely it is an oxidation reaction. During this oxidation reaction, there is a release of electrons. Yes. Electron release will occur by performing this chemical reaction. In this chemical reaction, electron release will occur. These electrons which are released, these electrons are picked up by the bacteria and taken up to the ETS, electron release. They will be taken to the electron transport system. And in the electron transport system, with the help of a complex, name of complex is ATPase. इस complex के through there is synthesis of ATP. Now this ATP which is formed, now it is utilized for carbon dioxide fixation. Carbon dioxide fixation होगा, जिसकी वजह से there is synthesis of food, there is synthesis of sugars. तो in this category of bacteria, food का synthesis हुआ. But for the synthesis of food, now light energy is not utilized. Now the energy is obtained from chemical reactions. So when the energy obtained for synthesis of the food is from chemical reactions, so this category of bacteria can simply be known as chemoautotrophs. So got it? Chemoautotrophs, instead of the light energy, they will be utilizing chemical energy. Now, after the nitrifying bacteria, one more example I will be citing here. That is iron bacteria. Iron bacteria, they convert ferrous salts into ferric one. Ferrous salts को convert करेंगे ferric salts के अंदर. Right? Again, there is oxidation. Oxidation की वजह से release होगा electron. Electron will follow the same pathway resulting in the synthesis of the food. So it is sure that for the chemoautotrophs, the chemical reaction which is involved, that is oxidation, which will involve in the release of electron. I'm citing one example here of bacteria, iron bacteria, that is Ferrobacillus. Next category, hydrogen bacteria, involved in the oxidation of hydrogen into water. Example here is hydrogenomonas. Last category, sulfur bacteria. Sulfur bacteria, they oxidize H2S into sulfur. H2S is oxidized into the sulfur. Bacteria involved here is Begetoa. Next, this sulfur, it can be oxidized into the H2SO4, formation of acid. Bacteria which is involved here, that is Thiobacillus. राइट तो यहां पर कोई भी कंफ्यूजन नहीं होना चाहिए. ये जो सल्फर बैक्टीरिया है, इट इज इन्वॉल्व इन द फूड सिंथेसिस बाय यूटिलाइजिंग केमिकल एनर्जी, कीमो ऑटोट्रॉफ. इससे पहले जो हमने सल्फर बैक्टीरिया पढ़ा, पर्पल सल्फर बैक्टीरिया, ग्रीन सल्फर बैक्टीरिया, दे आर इन्वॉल्व इन फूड सिंथेसिस by utilizing light energy. So they are photoautotrophs. So sulfur bacteria can be photo as well as can be chemoautotrophs. Now in this category of bacteria, yes, could you please tell me कि यहां पर जो इलेक्ट्रॉन डोनर है, इट इज ऑर्गेनिक और इनऑर्गेनिक कंपाउंड. यहां पर इलेक्ट्रॉन एंड हाइड्रोजन डोनर, organic or inorganic compound? Yes, here electron donor is inorganic compound. Bacteria in which electron donor is inorganic compound. Just now I have discussed this category of bacteria is known as lithotrophs. So they are also lithotrophs. So it means more appropriately they can also be known as chemoautotropic bacteria. Chemoautotrophs. Now after autotrophic category, now I will be dealing with the second mode of nutrition, that is heterotrophs.